There is a concept in nutritional biochemistry called the cellular environment, and it refers to the sum of the chemical conditions surrounding every cell in the body at any given moment. The level of oxidative stress, the concentration of inflammatory cytokines, the availability of detoxification enzymes, the quality of the DNA repair machinery, and the hormonal and metabolic signals that determine whether a cell grows, repairs, or dies. This environment is not fixed. It fluctuates with every meal, with every inflammatory trigger, with every exposure to the carcinogens and oxidants of daily life, and with the cumulative dietary pattern that determines the baseline condition of the biochemical landscape in which the body's cells operate. The reason that diet is relevant to cancer risk, to cardiovascular risk, to metabolic disease, and to the inflammatory conditions that underlie so much of the chronic illness of aging is not because any specific food is a medicine, or because any specific compound eliminates any specific disease. It is because specific compounds in specific foods directly support the biological systems, whose job it is to protect the cell from the damage that, over time, and accumulated in sufficient degree, produces chronic disease. The antioxidant enzymes that neutralize the reactive oxygen species attacking DNA, the detoxification enzymes that conjugate and excrete carcinogens before they form DNA adducts, the anti-inflammatory pathways that limit the chronic cytokine output that accelerates cellular aging, the vascular mechanisms that maintain the oxygen and nutrient delivery that healthy tissue depends on. All of these are supported or undermined by the dietary pattern, day by day and meal by meal, across decades of accumulated exposure. The 4 vegetables I am going to discuss today are not miracle foods. They do not cure cancer. They are not treatments for any condition. They are among the most evidence-supported plant foods for their specific activity on the cellular level biological systems I have described, through mechanisms that are increasingly well characterized at the molecular level. And for adults over 60 whose cellular environment has been accumulating damage over decades, and whose biological defense systems have been progressively declining, the specific support that these vegetables provide is proportionally more important than it was at 40. Before anything else, what I share in this video is educational information only. It is not medical advice. Dietary changes support health but do not substitute for medical evaluation, cancer screening, or treatment for any condition. What I want to give you today is the specific mechanistic evidence behind these 4 vegetables, so that your dietary choices can be grounded in what is actually known about how they support the body. I am Dr. Andrew Carter, and I have spent 15 years working exclusively with adults over 60. The nutritional environment of the aging body is one of the most modifiable inputs into the chronic disease outcomes that define this age group, and these 4 vegetables represent some of the most evidence-supported specific interventions available through the daily diet. If this is your first time on this channel, subscribe right now because every week I bring you specific, grounded information about what your body needs after 60, and this video applies the same mechanistic rigor to plant foods that we apply to medications and supplements throughout this channel. Let me establish the biological foundation before discussing the vegetables, because the mechanisms I am going to describe are ones we have touched on in multiple previous videos, and that deserve to be placed in the specific context of cancer protection and chronic disease prevention. Chronic disease, including most cancers and cardiovascular disease, develops through a progression that begins at the cellular level and advances over years to decades. Cancer specifically requires a cell to acquire multiple genetic mutations affecting growth regulation, DNA repair, cell death, angiogenesis, and invasion before it becomes clinically detectable. The probability that a cell accumulates those mutations to a sufficient degree depends partly on genetic factors outside anyone's control, and partly on the rate of DNA damage, the efficiency of DNA repair, and the cellular environment that either suppresses or enables the progression of mutated cells. The modern dietary pattern that most powerfully creates the cellular environment, most permissive to chronic disease development, is the ultra-processed food pattern, which drives chronic inflammation through its emulsifier and refined carbohydrate content, drives oxidative stress through its industrial seed oil oxidation products, and impairs the gut microbiome that supports both systemic immune surveillance and the barrier function preventing inflammatory microbial products from entering the circulation. We discussed this in detail in the dietary cancer risk video on this channel, where the evidence for ultra-processed food and cancer risk was presented with its specific mechanisms. The four vegetables I am going to describe today operate in the opposite direction. They reduce the chronic inflammatory burden. They activate the detoxification enzyme systems that clear carcinogens before they damage DNA. They provide antioxidants that neutralize the reactive oxygen species, attacking DNA and cell membranes. And they support the vascular function that delivers oxygen and immune cells to the tissues where the body's surveillance and repair work is done. They are not miracle foods and they are not treatments. They are specific interventions into specific biological processes whose adequate function is one of the most important determinants of chronic disease risk across a lifetime. The three most important modifiable inputs into this cellular environment are oxidative stress, which determines the rate of DNA damage, chronic inflammation, which both produces oxidative stress and provides growth-promoting signals to cells that have already accumulated some genetic damage, and the function of the body's detoxification systems, which determine how efficiently carcinogenic chemicals from food, environment, and metabolic byproducts are conjugated and excreted before they form the DNA adducts that produce the initial mutations. The four vegetables I am going to describe each address one or more of these three pathways with specific, documented mechanisms at doses achievable through ordinary dietary consumption. None of them is a guaranteed protection against any specific disease. All of them, consumed consistently as part of a broader whole food dietary pattern, contribute to the biological environment in which the body's natural defense and repair systems are better resourced. The fourth vegetable is spinach, and its contributions to the cellular environment operate through three distinct mechanisms that deserve individual explanation. The first is its nitrate content and the dietary nitrate pathway to nitric oxide that we have discussed extensively in the circulation and cardiovascular videos on this channel. Spinach is among the highest dietary nitrate vegetables available, containing 250 to 780 milligrams of nitrate per 100 grams of fresh spinach, depending on the variety and growing conditions. Through the oral bacterial conversion pathway, this nitrate produces nitric oxide that improves endothelial function, reduces vascular resistance, and enhances the oxygen and nutrient delivery to every tissue, including those under oxidative and inflammatory stress. The relevance to cancer protection specifically is through the vascular pathway. Adequate tumor microenvironment oxygenation is paradoxically relevant to the immune surveillance of premalignant tissue. Natural killer cells and CD8-positive cytotoxic T cells, the immune cells responsible for recognizing and destroying cells with abnormal surface proteins, including early cancer cells, are impaired in function in hypoxic, poorly perfused tissue. The nitric oxide-driven improvement in tissue perfusion from dietary nitrate supports the immune surveillance capacity of the tissues most at risk for malignant transformation. The second mechanism is the carotenoid antioxidant content of spinach, specifically lutein, zeaxanthin, and beta-carotene. In the context of cancer protection, these carotenoids quench the singlet oxygen species generated by UV light and metabolic activity that would otherwise produce the double-strand DNA breaks and base modifications that initiate mutagenic lesions. The Journal of the National Cancer Institute has published multiple prospective studies finding inverse associations between carotenoid intake and risk of several specific cancers, with the evidence particularly consistent for lung cancer and cervical cancer. The third mechanism is the folate content of spinach, relevant specifically to DNA methylation and to the 1-carbon metabolism pathway required for DNA synthesis and repair. Adequate folate is required for the proper methylation of DNA, and DNA hypomethylation in specific gene promoters is one of the early epigenetic events in carcinogenesis. Adults with low folate status show higher rates of chromosomal instability and DNA strand breaks, and spinach provides approximately 60% of the recommended daily folate intake per 100-gram cooked serving. Type in the comments right now how many servings of leafy greens you eat per week on average, because the gap between the intake associated with the strongest epidemiological protection and the typical intake in adults over 60 in most developed countries is one of the most practically meaningful nutritional gaps in this age group. The third vegetable is garlic, whose sulfur-containing compounds have accumulated one of the largest bodies of biomedical research of any plant food, across cell culture, animal models, and epidemiological studies in human populations. The primary bioactive compounds in garlic are the allyl sulfur compounds generated when garlic cells are disrupted, allicin from the enzymatic conversion of alliin by alliinase, and the further metabolites of allicin including diallyl sulfide, diallyl disulfide, and diallyl trisulfide. We discussed these compounds in the arterial morning drink video, in the context of their cardiovascular effects. In the specific context of cancer protection, these compounds work through a different but related set of mechanisms. The allyl sulfur compounds in garlic induce apoptosis, programmed cell death, in cancer cell lines through multiple pathways, including activation of Caspase cascade enzymes and disruption of the mitochondrial membrane potential that cancer cells maintain in their resistance to normal cell death signals. This effect has been demonstrated in cell culture across breast, colon, lung, prostate, and stomach cancer cell lines. The important caveat is that cell culture results do not directly translate to clinical outcomes, and the concentrations required to produce apoptosis in some cancer cell lines exceed what can realistically be achieved from dietary garlic consumption. The research provides mechanistic plausibility for the epidemiological associations, not a direct proof of clinical efficacy. The epidemiological evidence for garlic in cancer is most consistent for gastric and colorectal cancer. A meta-analysis published in the European Journal of Clinical Nutrition examined prospective and case control studies and found that higher garlic consumption was associated with significantly reduced risk of gastric and colorectal cancer, with the risk reduction most pronounced at consumption levels of 2 or more cloves per week. The anti-inflammatory effects of garlic allyl sulfur compounds through inhibition of NF-κB signaling and reduction of pro-inflammatory prostaglandin production are the mechanism most likely to contribute to the population-level cancer risk associations. Chronic inflammation is a driver of carcinogenesis at multiple anatomical sites, and the dietary anti-inflammatory activity of garlic, consistent across studies and plausibly mechanistic, is a genuine contribution to the cellular environment that reduces cancer promotion signals. The preparation detail that matters most for these effects is the same one we discussed in the morning drink video. Garlic must be crushed or minced and allowed to rest for at least 10 minutes before cooking or consumption, because the alliinase enzyme requires this time to convert the alliin precursor to a lysine, and heating immediately after cutting inactivates the enzyme before the conversion is complete. Garlic added to the pan immediately after cutting provides flavor but produces little of the bioactive allicin. And share this video right now with one person in your life over 60 who cooks with garlic but heats it immediately and has never known that the preparation timing determines whether they are getting the flavor or the biology. And if you are not yet subscribed to this channel, tap that button now and ring the bell. I want to tell you about Linda, 67 years old, retired scientist from Cambridge, England. She came to me specifically interested in the evidence behind dietary cancer prevention, following a family history of colorectal cancer in both her father and her brother. She asked me directly, which foods have the most credible evidence? She was already eating well by most measures, but when we examined her intake in detail, her leafy green consumption was 2 servings per week, her garlic intake was minimal, her broccoli consumption was occasional rather than regular, and she had never specifically thought about beet consumption. I explained the specific mechanisms and the specific evidence for each of the 4 vegetables I am discussing in this video, and we built a practical daily incorporation plan. 6 months later, she described her dietary pattern as the most intentional it had ever been, with each food choice now carrying a biological rationale that made it sustainable in a way that generic healthy eating advice never had. She told me, knowing why I am eating each food makes the choice feel like something I am doing rather than something I am supposed to do. I told her, the mechanism is the motivation. When you know what the sulforaphane is doing in your cells, broccoli stops being a boring vegetable and starts being the most interesting intervention available. The second vegetable is beets, and the mechanism of their cancer protective potential operates through the vascular pathway of nitric oxide, combined with a direct antioxidant and anti-inflammatory contribution that is distinct from the nitrate effect. Beets are the vegetable with the highest inorganic nitrate content of any commonly consumed food, containing 250 to 900 milligrams of nitrate per 100 grams of raw beet. Through the same oral bacterial conversion pathway we discussed for spinach and that we described in detail in the circulation drinks and arterial morning drink videos, This nitrate is converted to nitric oxide that produces vasodilation, improves endothelial function, and enhances oxygen delivery to tissues. In the context of cancer prevention specifically, there is a phenomenon in tumor microenvironments called the Warburg effect, the tendency of cancer cells to rely on anaerobic glycolysis for energy production, even in the presence of oxygen, producing lactic acid, and creating the acidic, low pH microenvironment that further suppresses immune function and promotes cancer cell survival and invasion. Adequate tissue oxygenation from the nitric oxide-driven vascular effects of dietary nitrate does not directly reverse the Warburg effect in established tumors, but in pre-malignant tissue, where cells are under metabolic stress, adequate oxygen delivery supports the oxidative phosphorylation that normal cells depend on and that cancer cells are increasingly abandoning. The betulin pigments that give beets their distinctive deep red color are a distinct class of antioxidants with specific anti-inflammatory and antioxidant properties that have been studied in their own right. Betinin, the primary betylane, has been shown in cell culture studies to inhibit the activity of several inflammation-related enzymes and to reduce lipid peroxidation in erythrocytes under oxidative stress. A study published in the Journal of Agricultural and Food Chemistry found that betinin reduced the production of reactive oxygen species in macrophages activated by inflammatory signals, with an effect comparable in magnitude to quercetin, one of the most potent dietary flavonoids. The folate content of beets, relevant to the same DNA methylation pathway I described for spinach, provides an additional mechanism for DNA damage prevention, and the fiber content of whole beets supports the gut microbiome with fermentable substrate for short-chain fatty acid production, including butyrate, which has specific anti-proliferative effects on colorectal epithelial cells that are relevant to the cancer-protective evidence for dietary fiber, discussed in multiple meta-analyses. Beet juice provides the highest nitrate dose most efficiently. Roasted beets retain the betylins and the folate, but with some reduction in nitrate content. Pickled beets, while preserving the betylins and some folate, contain added sodium that reduces their net cardiovascular benefit. Raw grated beets in salads provide the full complement of nitrate, betalanes, folate, and fiber. If you are not yet subscribed to this channel, tap that subscribe button now and ring the bell because every week I bring you the specific mechanistic evidence that distinguishes why each food in the category of cancer-protective vegetables is working and through what pathway, rather than the generic recommendation to eat more vegetables that most people have heard and not acted on. The first vegetable is broccoli and the broader cruciferous vegetable family: cauliflower, kale, Brussels sprouts, cabbage, arugula, and bok choy, all of which share the glucosinolate chemistry that produces the most studied plant compound in cancer prevention research. Sulforaphane, the compound produced when the glucosinolate glucoraphanin is converted by the enzyme myrosinase, upon disruption of cruciferous vegetable cells, is the single most intensively researched plant compound for cancer prevention at the molecular level. The research spans more than 3 decades, involves multiple cancer types, and has produced enough mechanistic clarity that sulforaphane-producing vegetables are frequently used as model compounds in basic cancer prevention research. The primary mechanism is the activation of the NRF2 pathway, the nuclear factor erythroid, 2-related factor 2 transcription factor, that we discussed in the mobility and age spots videos on this channel in the context of antioxidant enzyme activation and skin health. In the specific context of cancer prevention, NRF2 activation by sulforaphane drives the transcription of a family of phase 2 detoxification enzymes, including glutathione S-transferases, NADPH quinone oxidoreductase, and heme-oxygenase, that conjugate electrophilic carcinogens and reactive oxygen species to glutathione and other carrier molecules, enabling their excretion before they can form the DNA adducts that initiate mutagenic lesions. This is the detoxification pathway at the molecular level. Sulforaphane from cruciferous vegetables activates the genetic program that arms the cell with the enzymes most capable of neutralizing the carcinogens it encounters. A cancer cell that never acquires the initiating DNA mutation cannot progress to malignancy, and the sulforaphane-induced detoxification enzymes reduce the probability of those initiating mutations by accelerating the conjugation and excretion of the chemicals capable of producing them. The epidemiological evidence for cruciferous vegetables and cancer is among the most consistent in nutritional cancer research. A meta-analysis published in the International Journal of Epidemiology, examining data from more than 20 studies, found significant inverse associations between cruciferous vegetable consumption and risk of bladder cancer, breast cancer, colorectal cancer, and lung cancer, with the magnitude of the association consistent with a biologically meaningful effect rather than statistical noise from confounding. Beyond the NRF2 pathway, sulforaphane has been shown to specifically target cancer stem cells, the small subpopulation of cells within tumors that are responsible for tumor regrowth after treatment, through inhibition of the HDAC enzymes that maintain the stem cell program in those cells. This specific activity on cancer stem cells, which most pharmaceutical chemotherapies do not effectively target, is one of the reasons sulforaphane has attracted significant pharmaceutical research interest as a potential adjunctive cancer treatment, though this remains in early phase clinical investigation. The preparation principle for cruciferous vegetables is the same as we have discussed in previous videos. The myrosinase enzyme that converts glucoraphanin to sulforaphane is denatured by heat, so cooking destroys most of the enzymatic conversion before it can occur. One approach that preserves sulforaphane production is to chop or cut cruciferous vegetables and allow them to sit for 5 to 10 minutes before cooking, as the myrosinase has time to convert some glucoraphanin to sulforaphane before the heat inactivates it. Alternatively, light steaming rather than boiling preserves a significantly higher proportion of both the glucoraphanin and the myrosinase compared to boiling, which leaches both into the cooking water. Broccoli sprouts, the young seedlings of broccoli harvested at 2 to 3 days of growth, contain 50 to 100 times the concentration of glucoraphanin per gram as mature broccoli, making a small amount of sprouts an extremely efficient sulforaphane source. A tablespoon of broccoli sprouts provides the sulforaphane precursor equivalent of several hundred grams of mature broccoli. Growing broccoli sprouts at home requires only a jar, water, and broccoli seeds available from health food stores, and produces ready-to-eat sprouts within 3 days of beginning the process. The sprouts are consumed raw, preserving the myrosinase enzyme intact, and their mild flavor, similar to radish sprouts, blends readily into salads, sandwiches, and smoothies. For adults who find regular broccoli preparation burdensome, homegrown broccoli sprouts represent a lower volume, higher potency alternative that addresses the primary barrier to consistent cruciferous vegetable consumption. Here is the complete practical framework for incorporating these 4 vegetables into a daily dietary pattern in a way that provides their specific biological benefits at doses consistent with the evidence. Spinach provides its primary benefits through its nitrate content, its carotenoid antioxidants, and its folate. Approximately 200 to 300 grams of raw spinach, or 100 to 150 grams cooked, consumed 4 to 5 times per week, provides clinically relevant doses of all 3. Raw spinach preserves more folate than cooked, as folate is water-soluble and heat-labile. The nitrate content is similar raw and cooked as long as the cooking water is not discarded. Garlic requires a minimum of 10 minutes between mincing and heat exposure for full alliinase conversion to a lysine. 2 to 3 cloves per day, incorporated across meals, provides the dose range associated with the strongest epidemiological cancer protection evidence. Aged garlic extract is an alternative for adults who find raw garlic difficult to digest, as the aging process stabilizes some allicin metabolites without the irritant compounds of fresh garlic. Beets consumed as roasted whole beets, raw grated in salads, or as a small daily portion of unsweetened juice, contribute nitrate and betalains at doses within the range studied in clinical trials. The combination of beet consumption with spinach consumption provides additive nitrate from 2 independent high-nitrate sources. Broccoli or broccoli sprouts consumed 4 to 5 times per week, prepared with either the chop-and-wait method before cooking or with light steaming, provides the sulforaphane at doses associated with the epidemiological cancer protection evidence. For adults who want the most efficient sulforaphane source per gram of food, broccoli sprouts eaten raw, added to salads or sandwiches, provide a dose achievable in a single tablespoon that would require several hundred grams of cooked broccoli to match. 4 things to carry away. First, the cancer protective and disease preventive properties of these 4 vegetables operate through specific, documented molecular mechanisms, not through generic healthy eating effects. Spinach supports vascular oxygenation and immune surveillance through dietary nitrate, DNA methylation through folate, and antioxidant protection through carotenoids. Garlic inhibits cancer cell proliferation and reduces carcinogenic inflammatory signaling through allyl sulfur compounds. Beets support vascular oxygenation through nitrate and reduce oxidative stress through betalain antioxidants. Broccoli activates the detoxification enzyme program through sulforaphane and NRF2 pathway activation. Second, preparation matters as much as choice. Garlic minced and used immediately, without the 10-minute rest, loses most of its allicin. Broccoli boiled heavily loses most of its sulforaphane. Spinach cooked in large volumes of water and discarded loses much of its folate. The preparation details are not trivial. They determine whether the compound reaches the biological target or is eliminated in the preparation process. Third, the evidence for these vegetables is strongest and most consistent when they are consumed regularly, meaning multiple times per week across years, rather than occasionally or in large amounts periodically. The biological mechanisms they activate are adaptive responses that require sustained stimulus to maintain their effect, and the epidemiological protection associated with these vegetables in population studies reflects habitual consumption over years rather than short-term supplementation. Fourth, these vegetables support the cellular environment in which the body's own defense systems operate more effectively. They are not substitutes for cancer screening, for medical treatment of any diagnosed condition, or for the other lifestyle factors that most powerfully determine chronic disease risk, including physical activity, non-smoking, alcohol moderation, and healthy body weight. They are a specific and evidence-supported dietary contribution to the biological environment that makes those defense systems work better. Your cells are managing oxidative damage, repairing DNA mutations, clearing carcinogens, and suppressing abnormal cell growth every hour of every day, and they have been doing so for every year of your life. What you eat determines how well-resourced those defense systems are for the next round of that work. The 4 vegetables I have described today are among the most direct dietary inputs into that resource supply that the nutritional evidence currently identifies. Share this video with one person in your life over 60 who eats reasonably well but whose diet does not specifically include broccoli, garlic, spinach, and beets with any regularity, because the gap between a generally healthy diet and one that specifically includes the compounds I've described is a genuinely meaningful gap in the biological environment those compounds support. And subscribe to this channel, because every week I come here with one purpose: to give you the specific information that makes your dietary choices genuinely mechanistic rather than vaguely aspirational. Aging does not take away your body's ability to recover. It simply reminds you to care for it the right way. Take care of yourself.